Alright, I am about to shock you to the core. You see this? This is a brain scan using what they say their MRI, and they're colorizing these different areas in the brain that represent probably different, motor, your arm, your leg, or this or that. But they are, you see how intricate all of this stuff is here? All these different colors? Why? The only reason you see different colors is there because there's different materials there. There's different fibers. The coating of the fiber is different. The molecules are different. And I'm going to show you something. A brain scan next to a crystal skull. And this is going to blow you away. It's the Mitchell Hedges skull. Nobody ever realized it, but it's a real skull. Now, unless you have some idea about anatomy and all, and you have an open mind and you can look and stop and think, you're never going to understand that that is a real skull. That was from some real human person a long time ago. And they died in the Great Flood. I'm almost absolutely certain of it. And all of the silicon replaced literally everything. Now, the key with silicon is it will replace everything but the membranes. The membranes still show up a little bit different, and I'll show you. I have a bunch of them here that, well, I have some that I'll show you in a minute. But this is right here. These are all the different roots. It's exactly the same, identical, absolutely identical. Now, what is this? Why is this here? This one's way back here. Well, it was on its face. And I can tell that because it was laying on its lip and its nose. It's pushed back. You see the lip and the nose? The teeth, when I first saw this, I said, no, nobody's got teeth like that. Well, they did. Look at that. They're absolutely flawlessly perfect. I can't account for that. But we also have that little bone hole right there. And everything here was replaced with silicon. It invaded everything. And it just took it over. Now, I have a silicon heart here. Well, I have two of them, and one of them's cut. And I'll show them to you in a second. But this is identical to that. Now, take your time and look at it. All right, remember this. It's coming up this way, wrapping around and over. Now, this is way deep inside. Nobody carved that on the outside. That's inside. Just as this is inside your brain. Or mine, too. Now, this is that section of the brain is here. And it's all of its brain cells, which are, the neurons, feed all of those signals to wherever they're going. And anyway, it all comes down through the brain stem and goes to the whole body. Now, how it does that, I'm not 100% certain, but these fibers, there is a ton of them, and each one of them has all of that membrane activity going on. Let me see if I can find, well, here it is right here. Well, let me just read you this. In the previous chapter, we mentioned nerve fibers emanate from the nerve cells, which was that bundle of goo, and then there's nerve fibers come out from those cells. They're called neurons. Now, they're responsible for generating and carrying electric signals throughout your body. So it's like wires. As I showed you, they all have insulation around them. Neurons are present everywhere in the nervous system. So these are the neurons. The neurons are also present to receive the signals. Neurons are present everywhere. The brain, spinal cord, periphery, nerves, nerve fibers are all made up of axons of the neurons. Axons of the neurons. All right? Neurons have a unique structure that helps them do this. Examine the following diagram. Now, I think I have a better diagram. Hold on. Yeah, here. No. The neuron is the nerve cells, and those are in those bundles. like where you see a feeding into the fibers. Now let's just look at one of those bundles. I just don't want to miss anything. Don't miss a thing. All right, so this is all the neurons that are making the information, apparently, and sending it out through these literally like wires. Now, where all these other ones are going, I don't know. But this one looks like we can make a determination that there's a completely different jello ball here, basically, feeding out all those wires. So that's the way I'm going to take this. I could be wrong. Because something is feeding all these wires. They're not coming up here and dead-ending there. They're going to something to get a signal. I would think. What they're going to, I don't know. I can't see any ball, but this one I can. So that's why I'm working with this. Now let's see what happens as it comes out of here. Okay, this is going to be good for you to understand. When we talk about vitamin B12, it's known as cobalamin. It's water soluble, it dissolves in water. It's a vitamin, and it's involved in metabolism. Well, what is metabolism? Metabolism is when things get broken down. It's metabolizing. It's a set of life-sustaining chemical reactions done by enzymes. The three main functions of metabolism are to conversion of the energy in food to energy available to run cellular processes done by enzymes, the conversion of food to building blocks of proteins done by enzymes, to make lipids, nucleic acids, and all kinds of things. Everything's done by enzymes. And that's what metabolism is. You take stuff in, it's got to be broken down so you can use it. So B12 is one of 8 B vitamins, and B vitamins are just a class of vitamins. Now, what do they even consider a vitamin? Vitamins are organic molecules essential to any organism in small quantities for proper metabolic function. Essential nutrients cannot be synthesized, that means cannot be created. Essential nutrients, which you need, nutrition, cannot be made, which is synthesized, in the organism in sufficient quantities for survival. We can't make enough, they claim. Therefore, they must be obtained through the diet. So you have to have things when you eat things. So some of these vitamin supplements, who knows? But B12 is particularly interesting to me because of its extreme complexity. It's basically an enzyme. It's a coenzyme, and it's the most exotic of the B enzymes, of the B vitamins. Now, this I thought was interesting to think about. It's because it's a cofactor in DNA synthesis. DNA synthesis means you're building new DNA molecules. You have to have this. It's a cofactor. Now, vitamin B12 is the most chemically complex of all vitamins. Yes, it is. There it is right there. And for humans, the only vitamin that must be sourced from animal-derived foods or supplements. Only some archaea and bacteria can synthesize B12. So the bacteria can synthesize B12. B12 deficiency is a widespread condition, particularly prevalent in populations with low consumption of animal foods. This can be due to a variety of reasons, such as low socioeconomic status, ethical considerations, or lifestyle choices, such as vegans. And they say vegans can be in trouble without having this B12. My son is a vegan. He won't eat anything that ever had anything to do with life. I mean, it's just all plant stuff, and I applaud him for his ethical values, but it's kind of hard to live your life that way now. Okay, my friends, very exciting news today. If you've been following me at all, you know that I've been talking about how your gut, the bacteria in your gut, which create enzymes, are the only thing in your body that does any chemistry, and the chemistry is what your body runs on. And because of these enzymes are so unbelievably destructive to molecules, they can make and break things millions of times a second, where it would take years to do that if it wasn't for this particular enzyme, which allows it to create isotopes. I know this sounds confusing, but it will not be. And when I'm done talking to you today, it'll only take 15-20 minutes before you'll fully understand, and you'll be able to talk on a level of any researcher in this field, because I've done the research. I understand it very, very well, and I will make you understand it very, very well, or question me and we'll discuss it, because I think I understand what virtually all invasive diseases are. And now they're saying it's extremely simple. Look, Parkinson's link to gut bacteria suggest unexpected, simple treatment. Yes, unexpected, because they've never looked at it to understand the importance of the enzymes in the membrane. And it's, actually, this particular membrane has just been discovered a few years ago, so they're just starting to catch up. Let's see if we can make heads or tails of it. And this could be extremely important. And I'm telling you right now, I'm getting old quick. And Parkinson's, Alzheimer's, all those things, joint discomfort and everything, I'm starting to understand it, and I'm starting to actually, hopefully, intervene so it doesn't get bad quick. That's the best you can do. And now they're starting to understand. It's very simple to do. It's not a big, it's simply chemistry and enzymes. But the enzymes are only created if you have the bacteria. That's what I'm going to show you, the research. And I'm not telling you to do anything or not do anything. There's no medical advice given here. But you have to start by understanding the processes that run the body, and when they don't go right, what isn't right. And these people are suggesting to do exactly what I and my group have been suggesting for 6 or 7 years now, is to look at the bacterial products in a person, because that's what creates the enzymes. If you don't have the enzymes, you don't have the chemistry. So you start with the bacteria and you end up with the chemistry. Let's figure it out. It'll make sense. Well, okay, my friends, this really turned me on when I saw this about Parkinson's linked to gut bacteria. Suggested unexpected, simple treatment. It's gut bacteria. All right, now, this is what they're showing. This is the intestinal lining, I believe, illustration of colon epithelium lining, with all the bacteria and microbes and all that stuff floating around there. Now, here's what they're talking about. Researchers have suspected for some time the link between our gut and brain plays a role in the development of Parkinson's disease. Basically, the gut and the brain axis is regulated by bacteria creating enzymes. The enzymes are the catalysts that break down products instantly to make the chemistry, to make this reaction in the brain work correctly. If you don't have the chemistry, it just doesn't happen. So the gut creates the chemistry by using the bacteria. And I'll show you exactly how it works. It's very simple. So between the two of them, this guy here, the gut, says, I'm going to make all kinds of chemistry down here. If you need it to do a job, brain, I'll send it up to you through the fluid-filled highway. I'll explain everything to you, because that's what's in your body. Now, the gut makes a whole bunch of good stuff and sends it up to the brain. The brain does what it's supposed to do. Everything's fine. The gut doesn't have the good stuff to send up. The brain doesn't do what it's supposed to do. Things are not fine. So the gut-brain plays a role in the development of Parkinson's disease. Well, that's fine. Exactly how and exactly what, and then what are the chemistry? I want everything. I want the details. All right, this is how I do research, and this is what you should do if you want to do good research. Look at every single detail that they say. Here's what they're saying. A new study identified gut microbes likely to be involved and linked them with decreased amounts of riboflavin, which is vitamin B2, and biotin, vitamin B7. So these are microbes in your gut that are linked to decreased amounts of this and that, pointing to the way to an unexpectedly simple treatment that may help. Just B vitamins. Well, there's some microbes that are not there, that are not creating the right amounts of riboflavin and vitamin B7. This is incredible, and I think I have something to add. This is from Science Alert. I get all these links to all these kind of things. Parkinson link to gut bacteria suggests unexpected simple treatment. This is just today came out. Here it is right here. A new study just identified gut microbes likely to be involved and linked them with decreased riboflavin, which is vitamin B2, and biotin, which is B7, pointing the way to an unexpectedly simple treatment that may help. B vitamins. Well, why would B vitamins, just B vitamins, help you for this in your stomach? I mean, for your Parkinson's. And what's that got to do with B vitamins with a gut bacteria issue? Well, gut bacteria is what make all the enzymes. Well, guess what B vitamins are? They're coenzymes. All right, this is very interesting article. This is from Washington State University. It's nerve cell physiology. And it's chapter 3, and it's a little bit involved. Well, it's pretty involved, but you need to understand. Well, let me just cut to the chase here. They're going to go through, now that you have a sense of the nervous system. Well, this is chapter 3, so far we're just jumping ahead. But anyway, it says that we will examine the different properties of this signal, of the signals in the brain, as well as study how signals jump from one cell to the next. Some basic knowledge of biology and chemistry required to understand this chapter, which I will make it easily understood. So it may help to review if you feel rusty. Well, you're not rusty if you're here. You are unrusty. All right, this I thought was pretty good. This is from Ken Saladin, professor of biology emeritus, and he's talking about nerve fibers and the cross section. Look at this. You want us to talk about intricacy. Every one of these is a nerve bundle fiber, and inside every one of these fibers is a bazillion little fibers. And this is what they look like in actuality. Now, I'm going to get a closer up shot of that. But what it's talking about here is the nerve fibers in a nerve are enveloped in supporting cells called Schwann cells, which usually wrap around them in a spiral like a jelly roll, with the nerve fiber in the center. It's the insulation. In those cases, the wrapping is called myelin. It's a sheath. But in other cases, the Schwann cells only folds one around the other fiber, and then it goes more and more and more. But let's look at this. Well, I just have to focus it in here. You see what's in here? This is its one bundle of fibers. Now look closely at each one of those separate bundles, and then there's like a white fiber. Well, let me get a good picture. Hold on. All right, so hold on tight, because each one of these is a fiber. It comes out of a neuron, which is a nerve cell. and it goes to wherever it goes to control whatever it controls. And they are in these bundles, and this is what it looks like. I got some good shots of this. And these are the different colors of the different transition metal primarily, which is involved in the coating of each different type of fiber. Each one of them is going to have a different type of coating. And that coating is very important. That's the insulation. And this is so intricate, it's just unbelievable. The amount of enzymes, it staggers me every time I look at things. But I'm going to show you some of this in detail. And the first thing I'm going to show you is Mitchell Hedges' skull. There's no question whatsoever, that thing's real. You don't have to be a genius to see that. I mean, you just look. This is one of those brain scans showing the different fibers in the back of the brain here, and then coming up, all wrapping around. These different colors mean they're doing a different job, or at least they're coated with some different coating than the other ones. That's the way I take it, and I believe they're related to go into a certain area. But it is also the coating that's on there. So in other words, if you don't have the right coating on this green, maybe your arms won't work. If you don't have, if you have all the right coating on the green, your arms will work fine. But if it's not on the blue, you may have bad eyesight or whatever it is. Every one of these fibers controls something. And I mean, there's a bazillion of them. And yet inside of every one of those fibers is all of that intricacy that I just showed you. Let me just be sure we understand each other, because this is important. If you don't understand it, you don't understand it. This is what's in every one of those fibers. Every one of these is a fiber. You see the insulation around every one of those fibers? It's just nothing more than insulation. And they're in little bundles. I don't know if the bundles are going to go somewhere specific, or they just break into bundles here and there so they can flex. I don't know. But I have another shot of this. All right, so this is what's coming out of here. This is this. See these little bundles here? Those are those little bundles. Inside of each one of these is all of those. All right, and then they come out to the tips that go to wherever they go. But all of these have to be able to move around in your body, because these are nerve fibers. They go everywhere in your body. They're everywhere. So the insulation is just absolutely critical. And the only way the insulation can be good is if the chemistry is good. And the chemistry is only good if you can produce the chemistry. And the only way to produce the chemistry is enzymes. And the enzymes work in a specific way using isotopes. They're the only thing that can use isotopes, which means they can break molecules into half-lives 1,000,000 times a second. So you can go and build anything you want. So you can take the periodic chart. And right now they have no idea how anything could be bigger than iron. I'm not kidding you. But we use all these, and especially up in this range here, the transition metals, to move things in the body, because they're charged metals. Now, all of these above iron are made from isotopes, creating daughter isotopes moving up. All right, when something decays, it doesn't necessarily decay downwards. They can decay upwards. That's the key. They miss the upward decay. All right, so you have to understand the nerve cells. It's just like a wire coming out of here with a ton of different fibers inside of it. So this is an axon, this whole thing. It sends messages from cell body to muscles, glands, other neurons. So this is the conductor of the information. Now, the neuron, which is the nerve cell, which was that different colored blob in the back of the brain, feeds into these terminal branches of the axon, all of these different signals, and they come in those little bundles, and then they come out here, and it appears that they go to all kinds of different places throughout the body. All right, the nucleolus. So all of those fibers are coming down with all that stuff, and at the end, they hit something and do something. All right. I believe it's going this way into there. I'm not sure, to be honest with you, but it's a transfer of information here and there, probably both ways, I would imagine. But this is how complicated that is. And there is just billions of them probably inside of you. I don't know how many, but I would assume there is just staggering amounts. And every one of them has that insulation. You saw that? This is the most important thing to take away from this, is this insulation. And that requires a certain sheathing, which requires a certain chemistry. Let's go with that for now. All right, let me show you something. This is very cool. This is extremely rare to find something like that. I mean, just think about it. But I can tell you, there's no question, that was alive at one time. It died on its face and completely invaded by silicon in whatever environment it was in. This was found, I think, in South America. And they're known for all these kind of things down there. Now, I have a couple of crystal things here, which I will show you in a second. Not as spectacular as that, obviously, but I think pretty spectacular. And they're going to be hearts. So let's, again, if you don't see that, I don't know what I can say. Nobody could make that. There's absolutely no way. And nobody would push the lip back up like that, and they'd break a couple of teeth or something. I mean, who would do this so perfect? It's just, it is what it is. And actually, the jaw, it articulates off it. You can take the jaw out just like you take your own jaw off. You know, if all of the tendons and stuff were missing and the muscles, it just comes right off. Now, look at this. This is a heart. All right. Now, this also is a heart. You see the big thing right there? That's this. It's just twisted in a different direction. And then you have all the blood running down. And then you have this right here. I believe that's that stripe right there. All right. And this is some red blood. All right. It just shows you what a heart is like. And I believe that's where there's a, you know, it's one of the ventricles, or whatever they call it, one of the ventricle walls running down here, I believe. Because you got two sides to your heart. You got, it pumps in and it sucks back and boom, like that. Now, here's one cut across. This is how fast, you know, but by the way, this is all crystal now. That one there turned into crystal, just like that Mitchell Hedges. But there's so much fibrous stuff in the hearts. It's a lot of, it's all muscle and tendon and fibers and all of these veins and arteries. They're very, very, very tough, and they do not get replaced. The brain is easily replaced, and I can tell you right now that can happen in the humanity as well. You could fill that thing up easily, you could suck it out easily. That's what I've found. But these, there's so much fiber in there, there's so much pumping going on inside there that they took on the crystal, but it didn't fill up like empty cavities. This ended up being an empty cavity as the brain moved forward. You see it? And it just filled up with crystals. And that happens, like I got a lung here. Here's a lung. You see, this one here, the same thing happened. It was a cavity. Lungs have cavities in them. I got another one here that was completely voided. You see, they have cavities. That's the cavity right there. All right, and this one, everything avoided out of it, so it's just nothing. This one here is perfect. It's flawless. Except there's a big entryway, which they all have, and it just filled up with crystals, as they all did in this great flood, except here and there they just voided completely. Some of them preserved completely. Just depends. But anyway, this is how I learned all this stuff, is from the mud fossils. And this is what I can see, these actual fibers in my mud fossils. That's how I got onto this. So I can understand this as well as somebody doing an autopsy even better, because they can't see what I can see. Once you understand what you're looking at. All right, because even like everything I show, I can stand behind. Anyway, let's move forward on this. This is how detailed these fibers are. All right, I showed you this heart here. This is the thing right up there. That's the heart, and this is the stripe on the center there, which separates the ventricles. There's a lot of red blood, obviously, in the heart, and some of it just remained, but almost all of it transitioned into crystal. All right, but this is what the heart looks like. That's what a heart is, only this is sitting that way. Now, this is one cut open. You see here? You see all that stuff? It's the same stuff. These are all heartstrings and webbing inside the heart. All right, and this one again turned into crystal as well, and I just cut it open, and that is what it is. It's the same stuff. I'm going to show you a opal heart. Where do you see this? Okay, check this out. These are literally completely silicates. Very, very little coloration in here, which is a little tiny bit of the iron from the blood. All right, but other than that, there are silicates. Just like that has been completely washed of all of its stuff, and all it has is the structural material, and that's basically what we see here. And everything else has been replaced by clear silicates. Well, what if that heart was in an area where the silicates weren't clear, but they were flushed with blood? Because blood is transition metals. Let me show you something. Here's what blood is. All right, these are from blood. Your heart, I mean your blood is literally all metals. Every single one of these metals is in your blood, all the way up to uranium. They did a blood survey of like 26 different ones just to see, and they're all in there. And what they do is, because they have charges on them, they're transition metals. They carry things around your body, so they form colored compounds and complexes. And it's because they can carry, depending on the charge of the metal ion, and then what attaches to it. They take things and attach to themselves with these +3 and 4 and 2 and 5 charges. They carry them around and drop them off to somebody else. They give them something else. They return cleaned up. That's all you have to know about that, is the colors. Now, let me show you the colors of this heart, which is an opal heart, and it's just exactly the same as like this. If this was in solid blood and stayed in a certain type of chemistry, and I can't tell you exactly how this happened, but it was heavy in silicates and it was heavy in blood, the transition metals. And all of the different membranes in here, which there's a million different membranes, they all took on their own coloration of this to become stable. And that's what they want to do, is become stable. And they use the silicates and the transition metals to become solid, stable, like this. This just uses silicates. But here, there was all these metals, and they had no place to go, so they worked in with the silicates. All right, there it is. This is basically, if you cut this down the middle like that and opened it up just like that, this is what you have. This is called a Yoa nut opal. It's a heart. Now, I can tell you how it was laying and everything about this heart. It was laying this side down. So this was on top of that, laying like this. And I can tell you that because these are transition metals. Metals are heavier than the plasma, the raw red blood. And the raw red blood would have risen above, and the metals would have sunk to the bottom. That's what happened here. Now, you see how all of these different coloration on these different membranes? That's because different membranes have different chemistry. They want to attach with something that will bond with them, some of these transition metals that will make them stable, and that's what they did. Silicates seem to bond with just about everything, and if there's a lot of blood right there and it can't get away, it bonds with the silicates. And there's something in here where it was just watery silicates, and there's a certain chemistry goes on. I can't tell you I know 100% about it, but I can tell you one thing, it's no question whatsoever. This was heavy silicates, very clearish water other than blood. That's what I'm seeing. And whether this housed its own blood and turned like this, because it could, but still flushed with silicates, because it still has all of its membrane. You see, the membranes are still all the way around here. And a friend of mine did find a heart that still had blood in it. When he cracked it open, the blood was still open, and it was actually raw blood. Let me show you that. This is just stunning stuff, I know, but it's true. And until we understand that this is all real, you never understand these invasive diseases. They're invading the membranes that shield all these things. You know, I got a ventricle problem or whatever it is. It's an invasion of a membrane. The membranes coat. All of these things are coated. Even every fiber in your brain is coated. Every fiber in there has a coating. I showed you those cut across. You can see there's a million different fibers, and every one of them has insulation. And that insulation requires chemistry. Now let's talk about the chemistry. All right, you've seen the stuff, and you know, biology turns to. Well, look at this. This is a heart. This is an opal. On the other one's a heart. This is an eyeball. You see that? That's even the little connective thing that goes to your eyeball so you can turn it back and forth. And different materials in your body stabilize with a different transition metal. And these are pretty much in Australia. And I don't know, I really don't have a good explanation for opals, so I'll stay out of that realm. Other than I can tell you, like I said, they are bonding with these kind of transition metals, and that's in this area right in here. And those are the things that transition through your body. They transition in aqueous solutions. And that's how things are carried through your body. That's why you need, specifically, iron is the big boy. And that carries your oxygen around. And you're transitioning oxygen to like just pumping in and out like crazy. The amount of oxygen you use is just, it's incredible. And it's all happening in the body, and it requires iron and all the different levels of iron oxides, and they transition as they give up the oxygen to take oxygen on, and that just happens as a process of going through your lungs and so forth. Now I just want to point something out here. You see this fascially and that line coming in here? All of this is fascia, which is the coating of the fibers around the nerve fibers. It's the coating, the insulation, that's the fascia. And this is what I believe they also have a fluid-filled highway inside that fascia. Because basically all fascia has a fluid-filled highway underneath of it, like this does it too. That's going to be fascia, the outside fascia. They call it the peripheryum and the epinephrium. You know, different layers of tissue surrounding these bundles. But then in the bundles, they also have their own fasciae. There's blood vessels here and so forth. This is a very complex system. And they have to be able to move and do all these things, and they have to be insulated. So let's just think about it that way. I'm going to start with that. Okay, there is a lot to add to this, so let's get started. This is the simple, unexpected, simple treatment for Parkinson's. Is vitamins, B vitamins, they say. Let's just read it through. They suspect for some time that there was a link between our gut and the brain plays a role in the development of Parkinson's disease. So the gut, brain. A new study just identified gut microbes likely to be involved and linked them with decreased riboflavin, which is vitamin B2, and biotin, which is vitamin B7, pointing the way to an unexpectedly simple treatment that may be helpful. B vitamins. Now, supplementation of riboflavin and or biotin is likely to be beneficial in a subset of Parkinson's. That means a certain number of Parkinson's type people, disease patients, in which gut dysbiosis plays pivotal roles. If you got an issue with your gut, something's just not right. You're not digesting correctly. That's just a basic fact. And if you don't have the correct digestion, you're going to be either not getting something in, or you're going to be getting too much of something in. Digestion works both ways. It keeps stuff out and breaks it down, and also it breaks down food to make it acceptable to be absorbed. It also breaks down things that are trying to attack you and trying to break through your walls. And you've got a huge number of lymph particles inside of you to do that job, so nothing should be able to penetrate you if you have the right bacteria in those fluid-filled highways producing the right ribosomes, which I either showed you or will show you how that whole system works. The bacteria create the ribosome goes to the site, smacks it and kills them. All right, now this was something they were looking at from Nagoya University metal researcher Hiroshi Nakawaki and his colleagues wrote this paper. So I figured, well, let me look that up and make sure they're sort of legitimate. All right, he seems very legitimate. Here he is right here, Hiroshi Nakawaki. Now, gastrointestinal disorders in Parkinson's disease and other Lewy body diseases. Now, Lewy bodies, we'll talk about those. They're clumps. These Lewy bodies, the understanding of it remains elusive. Why is this happening? This is the National Institute of Health. This is the National Library of Medicine, in the USA. So this is not something that is just off the wall crazy people, although maybe it is, but it's what's mainstream. Now, let's look and see what he has to say. What is in this article about Parkinson's related to the gut disease? Because it's not just Parkinson's. It's virtually everything. If you look almost anything and look up attached to gastrointestinal disorders, you're going to see almost everything. It says one of the symptoms is da 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 da, and it will be gastrointestinal. Let's look up Alzheimer's. See, here it is right here. They got it totally backwards. Alzheimer's disease can affect, the Alzheimer's disease causes gastrointestinal tract in several ways, including gut inflammation, altered gut bacteria, and changes in cholesterol metabolism. Gut inflammation. It goes on and on about Alzheimer's disease causing that. No, Alzheimer's disease is not caused by it, I don't think. I think this is what causes the Alzheimer's disease. Very similar to what's happening with autism as well. All of these things are neurodegenerative. Something's happening with the insulation around your nerves, and it's bacterial. It's altered gut bacteria. We need to know who's got what gut is bacteria is good, and make sure everybody's got that same good stuff. Because there's going to be a whole plethora of different gut bacteria, but we don't know what they are now. I've been talking about this for a long time to get a database of what the normal gut bacteria is and why, when it gets altered, why is it altered? What's missing? What's too much? They have some of this, but they need a complete subset of understandings that when you bring your information in with what's happening to you and what bacteria are in you and in your blood and in your fecal matter, and that's what they're doing now. They're going for fecal matter and, well, I think they might have even talking about this in this new article. Hold on a second. But anyway, you can see it's gut bacteria. And that comes out in your fecal matter normally. All right, here's another report about autism, which is another one of these problems with the nervous system. Neurodegenerative diseases. Some studies report children with autism are more than 4 times more likely to develop GI problems than those without. The most common complaint are constipation, diarrhea, abdominal pain. Other issues can include all kinds of things. Reflux, nausea, everything. Okay, this is Harvard Medical School. And my friend Marguerite, who is trying to get this study underway over in Northern Ireland, had an autistic son that was, he was 9 years old, I think, 9 years old, I think, when we started. This goes back about 4 years ago or so, 5 years maybe. And at the time, he was so autistic that they wouldn't even take him into an autistic study. He was just off the spectrum, and he was going to be put in a locked ward, is the way they explain it over there. And basically going to padded cell for the rest of his life. Because he was banging his head, he was aggressive, he was all kinds of crazy things. He was not verbal, he couldn't handle going to the bathroom and all that business. It was, which is common in extremely severe autistic kids. Now, that's autism. And they found that the gut was a big issue, and now her son is doing pretty well. And this goes about 4 years or so, and she started this charity over there in Northern Ireland to try to get this research done to understand the gut bacteria. So anyway, that's another issue, but we're never going to get anywhere until they start to understand what the problem is. And here's Harvard. Now, as a matter of fact, Marguerite, maybe I already just said this, but she did go to Harvard Medical School for the ribosome stuff, to understand the nerve issues, because I worked with her for I don't know, a year or two, and then she went to took a course at Harvard in the nerve sheathing and ribosomes and all that, and we had a good time talking about it. But let's talk about long COVID. Here's another one. This is Harvard again. Can long COVID affect the gut? Emerging evidence suggests that GI problems may persist in some people who have had COVID-19. That gives you long COVID. Now, this is back in 2023, about a year ago. All right, this is really a fabulous article. This is by Harvard, Christopher Velez, MD. Now, COVID, we all know about COVID, so I don't have to go too deep into that. But here's the deal. It infects the gastrointestinal tract, esophagus, stomach, small intestines, colon. All of those linings must have some basic similarity that it can penetrate. They're saying it infects all of those things. Well, does it infect all of those things, or does it infect one of those things, but then it just spreads to all those things? Who knows? It can trigger abdominal pain, diarrhea, which often, but not always, clear up as the people recover. Now, then they talk about long COVID down here. And he says, few people, even in the medical field, are aware that long COVID symptoms may include chronic diarrhea and abdominal pain. Almost always they do. There's some gut dysbiosis. Something in your gut is not working right. Your digestive system's not working. And if it's not working right, the end product's not going to be normal, like a nice sausage like poop on a regular basis. And anyway. Why might the gut be involved? And then he goes through all of this stuff about it's not clear why chronic gut symptoms might occur with COVID-19. One possible insight is a well-known symptom called post-infectious irritable bowel syndrome. They know about this post-infection of just about anything, because an infection is caused by your body being attacked by something that you don't have the bacteria to fight back with. So one possible is well-known syndrome, well-known, post-infection irritable bowel syndrome. It may occur after a stomach flu. Any kind of flu, you're going to have irritable bowel because you don't have the right bacteria anymore. And especially when you take antibiotics and all these different things to combat these biotic invaders, well, they don't always just kill them. There's other casualties, which are the good ones. Anyway, they just say the nerve network of the gut is so complicated, sometimes called the second brain. When the nerves are working well, you won't notice a thing. You eat without pain. You move your bowels with ease. You have no GI worries. But if the nerves are not working well, then even the process of digestion remains normal. Then, even if the process of digestion remains normal, you may frequently have symptoms like pain or a distressing change in your bowel movements, such as diarrhea or constipation. All of that is because of the bacteria. It's all because of bacteria. Basically, that's what happens for digestion. The bacteria does the digesting. It's really as simple as that. And once known as functional GI disorder, these health problems are now called disordered gut-brain interactions. Viruses and bacteria infect the gut. Experts believe they may prompt a change in gut-brain signaling that can cause this irritable bowel syndrome to develop. Yes. Let me show you what does happen in your gut and why the bacteria is the key to everything. If you don't have the right bacteria, you're done. And and really, stool samples should give you a pretty good profile of bacteria. You see this? B vitamins act as coenzymes, which help enzymes speed up the conversion of chemical compounds. It's absolutely amazing how fast they work already, millions of times a second. These help speed them up. So B vitamins sound pretty impressive to me, if that's exactly what they do. And this is exactly what they say. B vitamins might help, who knows? Now I'm going to look up riboflavin and biotin. Those are the two things. But virtually all your diseases have issues that relate to gut. All right, and here it comes down to the end. He says we could perform gut microbiota analysis on patients to see what's wrong with their gut, and they should, because it's fecal. They use fecal metabolite analysis, yes. That's what he said, the author here. And he says, using these findings, we could identify individual specific deficiencies, administer oral riboflavin and biotin supplements to those with decreased levels. Well, that's one way, potentially creating an effective treatment. And a more effective treatment would be to replace the bacteria that does that job, because there's going to be a bacteria that creates this riboflavin and biotin molecule. In some manner or other is required in the process of stepping through the creation of these molecules. And without that bacteria, it's not going to work. Now you could supplement, yes. And that's what's happening with a lot of these probiotics. They're supplementation, and then there's nothing wrong with that at all. They're good, but the better way is to reintroduce the fecal matter. Transplant, basically, that's what appears to be the best way to re-establish the microbiome. And unfortunately, it's not being looked at as closely as it should. Because it's, this is fecal matter. But this is how you test what people have in their bodies, is to test their fecal matter. You conduct fecal matter metabolite analysis. Let's see, because that's what they can find. This is where these bacteria show up. They're all through your body, and they just go out with all the poop. If you got them, if you don't have them, they're not going to show up there. And that could be an issue. And that happens. I tell you what happened to me is I had a root canal done. And when they did that, they gave me antibiotics and just said, oh, just take these just in case you get an infection, it'll stop it. So I just took them like an idiot. And, and I'm not saying you shouldn't take them, but I never took any probiotics. And I had 2 surgeries. I had an anal fistula twice until a doctor told me, he says, take some Metamucil and Well, I'm not even going to tell you what he told me. But, because I don't give any medical advice. I don't give any medical advice whatsoever. I'm just telling you, this is what they're saying in this community about these situations. And they never took into account that layer. Even Harvard said they don't know about this. They don't understand this yet in the medical industry. See, this is Harvard. Few people, even in the medical field, are aware that long COVID symptoms may include chronic diarrhea and abdominal pain. That's even in the medical field. And I'm going to tell you something right now. Somebody very important to me has cancer. And I said, talk to your doctor about probiotics and so forth. Which there was a study about mice having a good cure rate while taking some of these probiotics. So she talked to the doctor, and the doctor said, well, you're not a mouse. That's what the response was. You know, like, yeah, do whatever you want. You're not a mouse. That's not right. Because I'd be very, very surprised if the doctor understood the enzymes and the membranes. It's invasion of membranes. Every cancer is an invasion of the membranes. And all of the membranes in your body that are of a certain type, like lymph nodes, they're going to be protected by the same bacteria that's floating around in the same thing. But if you don't have the bacteria, there's no protection anywhere. So it spreads. You know, it has to find a place it can invade first. And you may be on the edge of being able to resist it a little bit. But these coenzymes, I like the sound of that. That sounds very interesting. I'm going to look into that deeper. All right, anytime you talk about synthesis, it means you're synthesizing, you're creating something, you're making it. Biosynthesis just means that it's the generation of natural products through enzymatic reactions, the enzymes. Now listen to this. As in cellular metabolism, successive enzymatic reactions by a number of enzymes are generally required to achieve a single biological active compound. So you need a whole batch of different enzymes. So you need a whole batch of different bacteria. And they all work together to create one active compound which your body needs, whatever that might be. But that's why I say they work so fast that they can work on the half-life of these isotopes, because that's what it's all about, the half-life isotopes. And that, you have to understand, is all about dipole flood theory of the atomic model, which means that all the, hold on. All right, here's the key right here. Biosynthesis means the generation of natural products through enzymatic reactions, not only through just one enzyme, but it's successive enzymatic reactions by a number of enzymes are generally required to achieve a single biologically active compound. So to get some compound in there that does some chemistry in you, it requires a successive number of enzymes, just like that, and at the end you come out with the product. You know, I got to be honest with you, I feel like a fool. I passed right over vitamins. I never even looked at vitamins. If I hadn't seen this thing today about B vitamins, I'd still not be thinking about them. But these are water-soluble vitamins. Look at all the things they do. Methylene bridge. Methylation is how you turn genes on. They got all kind of different things. They're needed for protein, carbohydrate, metabolism. That means breaking them down. Red blood cell formation, certain brain chemicals. Folate, a vitamin needed to form red blood cells. This is for energy. Vitamin B. This is cyanocobalamin, the most chemically complex of all vitamins. B12 is essential for blood cell production. Listen to this. Nerve sheaths. Nerve sheaths. B12. That's why they're recommending B12. And proteins. It's also involved in the cellular metabolism of carbohydrates, hydrates, proteins, and lipids. It sounds like it's a pretty active thing. Now, B vitamins act as coenzymes. They boost the speed of the enzymes. It's just phenomenal how fast the enzymes work, where you boost them with these, and when they need a turbocharge, off they go with the B vitamins. Sounds like to me. All right, I'm just going to have to ask you to go back to some of my earlier videos I just did within the last week or so. It talks about these cells, and every cell in you, this is an animal cell. Every single cell, even your red blood cells, have these membranes around them, and inside they have all these little organelles, and here is where the ribosomes are, and here's the lysosomes. The lysosomes break open the ribosomes. The ribosomes create all of those proteins or enzymes, and here's what they look like when those enzymes open up. Here's what happened. You have the bacteria. It creates the ribosomes. The ribosomes come out of the bacteria wherever it carries it to. Now that ribosome, when it hits what it sees is something that bothers it, it tickles the little feelers on the ball, and it unsheaths. So it comes in like this, and then it goes, and it kills the thing that it bumped into, and then it just goes around killing all the other ones, because there's going to be a ton of them. There's not just going to be one, and it just keeps killing them until they're all dead, and then it gets consumed by itself. But they don't go away. They're deadly. They're like executioners. Now that's an enzyme, which is a destroyer. That's a protein, which is a building block. I've shown this a million times, and I have a whole batch of videos on this just recently, so I don't see any reason to get crazy. But here's the enzyme. When it gets next to chemistry, it destroys it because of all these little magnetic pulses. Bip, done, bip, 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 bip. It goes right down the line. And here's how big these invasions get. See this? I believe these are the ones triggering their balls to smack open and kill all of these invaders. I believe that's what's going on with this one. Could be the other way around. I don't know. And this is the whole process. Well, you got to go back through the other videos, because it's as simple as this. Ribosomes are programmed or program mRNA strands, and the ribosome is programmed by polypeptide chains, which come out of the bacteria, and the bacteria create the polypeptide chains through a program in their DNA. This programs the mRNA. The mRNA goes out and programs our RNA. So here's the whole process. It makes the ribosome code. It comes out, it constructs a ribosome. All right, the ribosome, because from the code, all the different molecules that are in a line, is just nothing more than a program code. That's all it is. And it's very sophisticated, because every one of these has up to 20 or 28 different possibilities. So it's a huge program. So it programs the mRNA, which is here, messenger ribonucleic acid, which is a single. The DNA is a double. And it breaks into it using RNA polymerase, which is another enzyme. This is an enzyme too, which creates the ribosome, and that's transferase. And this is polymerase. Any A's is an enzyme, basically. And it breaks right into the DNA and makes us immune, or can break in and kill you. All right, so don't forget, there's a whole lot of things going on here, but the whole end result is you have to maintain the correct environment of bacteria, which does all the work. They create the enzymes. Without the enzymes, nothing happens. And the enzymes work so fast, it's just beyond belief. Up to millions or billions, actually, sometimes, per second, they can create things that would take years to do if you didn't have the enzyme. These enzymes are so elegant, as you have seen. That didn't just pop out of nothing. And if it's not exactly like that, it's what's called denatured. It won't work. So if your enzymes are damaged for some reason, you don't have them, well, nothing's going to happen. You can't create the products you need. It really, I've said it hundreds and hundreds of times. But, this is not any medical advice. This is just research. What enzymes do we need? What creates those enzymes? What bacteria are involved? They're starting to look into this a little bit, to which bacteria cause which end products to be good or bad. And hopefully we can target those bacteria, either get them out or put them in, whatever you need to do. And he's exactly right, this researcher from Harvard there. We could perform gut microbiota analysis on patients and conduct fecal metabolite analysis and figure out what's wrong, what are they missing or what do they have too much of, and then address them in an individual manner. All right, I love you all. We need to get this done. And it's probably going to require some kind of funding, not by me. I have no money for this. But they're trying to get something going over there in Northern Ireland. And all I do want to be is part of the research to see how this thing plays out, because I think there's very simple solutions to a lot of devastating diseases. If you don't have the right products in your body, you are not going to be healthy. If you are attacked by something and you don't have the right bacteria to create the enzyme to fight back, you are going to be invaded. Simple as that. And every part in your body has a different chemistry. Your kidneys are different than your liver, is different than your gallbladder, is different than your lungs, your heart, your pancreas. Everything has a different membrane around it, because some of them are acid, some of them are caustic, some of them are who knows what. But the membrane is not going to be the same membrane as another one. So if this membrane doesn't have the right stuff, it doesn't work. If this membrane doesn't have the right stuff, it doesn't work, and this one does work because it's got its own right stuff. What we're dealing with Alzheimer's and Parkinson's and all these neurological disorders, autism, is membranes of the nervous system. They have their own chemistry, I'm sure, and they have their own insulation values and their own conductivity values, and that requires a balance of gut, a balanced gut. You can't have digestive system issues and have all that stuff work right. It's just not going to be. You could be depressed, you could be anxious, you could be nervous, you could be all kinds of terrible feelings in you. It controls every single thing in you. Is the enzymes control 100% of what happens in you is chemistry. It's chemistry. All right, I love you all. We'll talk later. Okay, this is an absolute stunner. I just came across this. These are the ribosomes. We all know about those. They get squirted out from the bacteria. And what do? They turn into these. They unsheath when they bump into something that they want to destroy, and then they become enzymes, very exotic particles. But normally they're like this. But when they hit something that tickles them, they just go, and then they attack and kill or break down, which is an enzyme, is a breaker. Proteins, which come from the same source, but they build. Okay, so they say everything's a protein, call it what you want. These are enzymes, they break things, proteins build things. That's the way I think about it. And they all come from the ribosomes. And the ribosomes all come from bacteria. There's no other place to get them. You cannot get that enzyme without that bacteria, without that ribosome. That's the most important thing to remember. And what does it look like when it unfurls and it comes out of its sheath and does all this stuff? It looks like this, and then it goes around just killing bazillions of other particles that instantaneously, I mean millions of times a second. So I said, well, enzymes are the source of all breakdowns. And, enzymes are the whole key to everything, and they're all produced by bacteria. And I just sort of left it at that. Now, I never even considered vitamins, but guess what? Vitamins are enzymes. That means they work with the enzymes. So I started looking at the chemistry of the enzyme, of the B12. Look at this sucker. That's got cobalt as the central core of the metal core, it appears. And these are all ligands attached to it. That's the way I'm looking at this. This thing is very, very complicated. Now, all the other B vitamins are, kind of simple. In other words, they can be as simple as this. They can be as simple as that, or as simple as something like that sticking off to the side and whatever. But when they get like that, that's almost an enzyme. As far as I'm concerned, that's an enzyme. Now, what it does, I don't know. I have no idea. But I can tell you what, you got a lot of hydrogen and nitrogen. I'd have to look this over, but it's mostly hydrogens. Carbon, hydrogen and carbon. You got some oxygen here and there. But I don't really know what that molecule does, but I can tell you what, that's the core. That's the metal core. And cobalt is one of the transition metals. You see it's right there, cobalt. And transition metals, as I think I have shown you, are these, somewhere I got them around here, are these. See, cobalt is right there, one of the transition metals. And what it is, it sits in the center and it's surrounded by the ligands. And in this case, it's really surrounded. I mean, you don't get any more surrounded than that. That's the core right there, cobalt. It's got a plus charge to it. I don't understand that much about this particular molecule, but as far as I'm concerned, that's an enzyme. That has a very large amount of chemistry going on there. Now, as a coenzyme, it may work with all kinds of different other enzymes, maybe not just one specific enzyme. It might be like some kind of a switch. I don't know. I just don't know. But I do know the enzymes that come out of bacteria are some very spectacularly complex chemistry. That's the kind of chemistry we're talking about. Now, you saw that coenzyme, you know, it might be like this part right here. So it's just maybe a piece of an enzyme. I don't know. They call them coenzymes, and they're vitamins. And I just didn't pay attention to them. So shame on me. Also, I mentioned before that everybody worries about the blood-brain barrier, and big things can't get through there. Like even an enzyme in that little structure like this is too big to get through the blood-brain barrier. There's a little spot in there that says, no, you're too big, you got to stay out. Well, when it's in its ribosome form, when it's in the ball form, it's small enough, it goes right through. And they collect inside the cells. And it looks to me like they have their own little space they occupy here inside of cells. This is an animal cell. I don't care what kind of cell it is. They have all of these different organelles in every single cell. They have mitochondria that makes energy. They have a nucleus. They have all these different Golgi apparatus, and all of these things are inside of every single cell, and they all have a membrane, and they have ribosomes that sit in there. Now, when those ribosomes are needed and they go out, they could be just hanging around in the brain. As far as I know, they must collect in these areas, collection points inside of cells, and then when they're required, they pop out, or I would say they pop out and go attack whatever they're supposed to attack. And when they do, then they get big. They come out and then they go bada boom, like that. Otherwise, they couldn't get through the brain barrier to get up here. That's the only way I can think. They come up as ribosomes, which is the little balls, and then they pop out when they're needed as the big enzymes like this. That just can't get to the brain. So there's a lot of little bits and pieces here. But the key is, if you don't have the bacteria, you're never going to get there. I don't care what happens, you're just never going to get it. So if you need that enzyme, you better have the bacteria. Well, I think that's about the best I'm going to be able to do for today. But these are the different types of bacterial shapes and arrangements. You know, they got the rod-like, they got curved, you got all these different ones. And inside of all these different ones, more than likely, are different bacteria. Almost certainly. I mean, they may share certain bacteria that are common to all of them, but if they are different looking, they're virtually certain to have different bacteria living inside of them. And you can get affected that, but maybe not by that. Maybe that'll kill you, maybe that'll cure you, whatever. This needs to be looked at in sort of a really research-oriented way. And I'm sure they must have done this, but I can't find much about it. I really haven't looked hard, so they may have some very good studies. But I would like to know, like we know that there's certain there's flesh-eating bacteria, strep A, and you can just eat through human flesh if it's not stopped by something. So we need to know what stops it. Something stops it. Now they can give you penicillin, I believe, for strep A. It's penicillin, and it's like a broad spectrum killer of biology. Now that's, I don't think, going to just kill strep A. So that's the problem with pharmaceuticals, is sometimes they're sort of broad spectrum. So you're much better off if you've got your natural immune system working, which requires the correct commensal good bacteria. And now they're starting to understand that, which create the enzymes. And now I started to understand the coenzymes make the enzymes function even better. And then they use the half-lives. Let me just show you that half-life thing quickly, because people don't understand how molecules break down, and you cannot understand it until you realize molecules are made out of literally hundreds of thousands of these particles, and when they break down, they can instantly change into other molecules using isotopes. But you have to have enzymes to break those down that fast, otherwise you could never do it. Half-lives in these things are nanoseconds. That's billionths of a second. But these things, enzyme go, done. Millions of things happen, and now it's a new whatever it is. So it comes along, and an enzyme or whatever, something comes along and hits it to tell it to create into a new thing. And it just, it's like food coming in. An enzyme comes and it goes bang, and it goes, done. And it's whatever you need to turn into, which is probably going to break down starches and fats and all that stuff. Anyway, these all have certain functions in biology somewhere in the universe, and some of them can really harm you, and some of them, I'm sure, probably have beneficial effects. I don't think we know enough about bacteria at the moment. And certainly we don't know enough about the amount that's in the human body and where it is and different types and what happens when you don't have it. I can tell you right now, bacteria is a gut issue almost exclusively, although you can get skin infections and so forth, but you have to keep up with general hygiene. Let's just understand that. But then on top of that, your body creates the chemistry to protect you. I'm telling you, it never gets old. It just never gets old. This is from the National Library of Medicine, again, relationship between vitamin B12 and cobalt, metabolism in domestic ruminant, and update. That means animals that have like digestion where the bacteria grows. It's ruminants like cows. All right, it's all finally starting to make really good sense. Cobalt is what we're talking about here, is a transition metal. Now, I was always talking about transition metals moving things back and forth, and I always thought of a transition metal being this, having a metal core. Yes, one of these is in the metal right in the center, and then they have what they call ligands. Now listen to what it says here. This is aqueous solutions. That means they're in watery solutions, which is your blood. It says these transition metals form colored compounds and complexes. The colors vary depending on the charge on the metal ion. These are the metal ions. And the number and type of groups of atoms called ligands. These are the ligands attached to the metal ion. This is the metal ion. In aqueous solutions means watery, bloody. The ions form complexes with the colors shown to the right. So I'm thinking, yeah, you know, you got a metal ion, yeah, you got a couple, a little this, a little that, they drag it around, no big deal. Come back, get some more, give and take type of thing, which is what they do. They transition molecules from wherever they are in the blood to wherever they need to go, and they pick up whatever needs to get out of there, and they bring it back to get it cleaned up in the liver and so forth. So this was always my thinking of a transition metal. I never saw anything like the B12. I got to be perfectly honest with you, and I take full responsibility for being an idiot and not even looking at vitamins. I just glazed over them thinking they were just nothing other than little mineral things. But now I realize, no. These are basically the transition metals, and this one here is B12. Look at the complexity here. You see that? This just doesn't get any better. I'm looking at B12, and they're talking about cobalt as being the metal core and having all these ligands, which it is extremely elegant molecule. Now, so I look up naturally occurring cobalt. It has 28 radioisotopes. Well, an isotope, I'll show you what they are in a second. And then these are the half-lives, how long they're supposed to last. Well, an enzyme can make that half-life, instead of 5 years, it can make it into 5 seconds or 5 billionths of a second. It's just amazing how fast they can create half-lives. And why is that important, Roger? Well, you got iron here. You got cobalt here, you got nickel here. All right, you see the three of them? Science right now has no idea, none, how you could get any of these heavier elements than iron. That's as high as they can figure out is iron was crushed together and made into iron. Above that, they have no mechanism for this. I'll tell you what the mechanism is from up bump. It's from bumping the iron up. And we can bump the cobalt up, we can bump the nickel up, we can bump the copper up, but it requires enzymes to do that, to be able to work so fast that they can go and it's done. And I mean, it really literally is that fast. And it's using all of these half-lives. You see, you have to understand dipole electron flood theory. It's not just 27 protons and 20 neutrons together, and big balls, 20 and 27, but it's thousands and thousands here. You're talking 47 times 2,000. Right, so it's 100,000 or so particles in this cobalt, but they're all cobalt. They say they all have the same number of protons because of its charge, apparently. However, the mass is bigger, 47, 48, 49, 50, 51, 52, 50. What does that mean? It means that the cobalt down here is like this. And it's got 27 and 20, they say. Well, and for me, it's got, let's say, 10,000. And then the next one down, you throw another one in, and it's 12,000. And then another one is 14,000. Another one is 16,000. And every time you bump one up, it adds another 2,000, approximately. So what is happening? We're down here, and if we decay this particle down here, and it gives anything off at all, it ends up being iron. That's the decay, is iron from cobalt. All right? So we know we can go down from cobalt down to iron. Can we go from cobalt up to nickel? Yes, we can. because cobalt gets bigger and bigger and bigger and bigger and bigger. It's still cobalt, 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 cobalt. This is where the stable cobalt is. So right here, supposedly, at 27 and 32, it does not want to give one up. It doesn't want to take one on. It's stable, and it will never have a half-life, supposedly. Now, and the half-lives are here. See these? Half-life. This is nanoseconds. That's less than 35 billionths of a second. Microseconds, nanoseconds. So these are how long they last in that configuration. So they can go, but you got to have enzymes to make them do that. All right, so the enzyme's the key. The enzyme is the key. The only thing that makes the enzyme is the bacteria. Basically, that's it. Now, we can go all the way from cobalt down to manganese. You see, that's another daughter isotope. But mostly the daughter isotopes are going to be the next one down, and then past stability, they're going to be the next one up. Watch, at stable, we hit a number of protons, a number of dipole electrons that it's just comfortable. Now, what happens above that? The daughter isotope is nickel. That's above cobalt. How did that happen? Because they have no idea how they get past this. Well, we've already passed from iron to cobalt to nickel. All right, so now our daughter isotopes are nickel. And don't forget, the enzymes are going, done. And I mean, 100,000 processes instantaneously are done because of this. It's called click chemistry. It's all it is magnetic fields going, done. I know it sounds crazy, but it is true. See, all this is nickel, 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 nickel. We're giving off nickel. We just made nickel from cobalt. But it takes an enzyme to do that, to break it up. It's not going to happen by itself. And don't forget, these are isotopes we're talking about. These are fractions that are missing or extra fractions on certain atoms. And that's what makes them unstable and giving things off or wanting to take things on. And because dipole electron flood theory says it's not a big proton like that, it's 1823 dipoles, not a big proton. The chemistry they show is so, well, look at this. Look at this. Now, think about what I'm talking about. You got hydrogen, just hydrogen. It's almost 2,000 particles. And then you hook it up with all these other things, oxygen and nitrogen and carbon and all that stuff, with the core of cobalt. You got literally at least millions of particles here, at least millions. And they're showing, I don't know, a couple of hundred or whatever it is. All right, this is a lot of chemistry to work with. So you need vitamin B12, it looks like to me. If that, they're saying it's important for a lot of things, and especially, I believe, the sheathing on neurofibers. And that's why they talk about it being good probably for Alzheimer's, Parkinson's, and all that sort of stuff, because those are neurodegenerative diseases. And there's some recent research that say they call them Lewy bodies, which are nothing more than clumps, I believe, of plaque-ish material that's up in your brain. It doesn't get swept out. Now, this is pretty exotic chemistry. Is that the sweeper? I don't know. But it's a coenzyme, so you're going to have other enzymes that are going to be trying to do that job and just not able to do it, it appears. Because it takes 18 years, 20 years, they say, and bills and bills and bills. Now, so something's doing some work. Something's happening, but it's just not enough to keep it going. And as you get older and older, apparently you lose some of that bacterial flora that's, really good. As far as I'm concerned, it's all bacteria. And also, I'm going to tell you right now, bacteria evolve. They have to. There's no question whatsoever. They evolve. They evolve their DNA all the time. Because anytime you create a new enzyme, you have to have a new bacteria. In order to have a new bacteria, it has to have new DNA. So that bacteria is fine. It's doing a great job of spitting out this ribosome. And that ribosome looks just like this once it unfurls. And that's perfect. Does the job exactly what it's supposed to do. But then all of a sudden, we get attacked by who knows what. And something happens in this DNA. It says, whoa, I saw that guy and I saw a vulnerable spot. If we added one of these things sticking off just like this, we can go and kill that guy. And somehow it changes his DNA to say, when you spit out of here, no more of this one. No, we're not, I'm just talking. A new bacteria has to come, or it has to reprogram the DNA. There's no other way, because the ribosome is a product of that DNA program. So now it squirts this one out, and it can go and attack something. And it does that by programming the RNA. As I think I showed you this, but I'm going to do it one more time because this is important. These are where these little stripes come out of here. All they are is balls like this come out, and they come up through here, and they come into what they call transferase. And they program messenger RNA into a ribosome. You see that ribosome? That ball is a ribosome. And this is the program. They programmed it. Now it comes out and it adds into our DNA, and it does whatever it does to our DNA. Whether it makes you sick or makes you healthy, a lot of this stuff is up in the air at this moment, but this is how you do get immunities, and this is what they do for vaccines. They give you an mRNA strand that supposedly has a sequence that attacks the thing that's attacking you. That's what it's made to do, and they appear to work pretty good as far as I'm concerned. A lot of people, oh, this and all that. Well, I've had everything, and I've never had a problem. And everybody I know has had everything, and they never had a problem. So, now look at this. This is the good guys against the bad guys. Now, I believe these are the good guys, and they're coming in to attack against these viral guys that are sweeping in to try to take over their guy's body. And these things will pop open and actually consume these or kill them, one or the other. I don't know which. I believe that's what's going on here. But it's some kind of bacterial attack, and somebody's counterattacking one way or the other. All right, see, they're calling these superbugs. Well, yes and no. They are evolving to be more aggressive, let's say. But I think we're backing off on our defenses. So if we can up our defense mechanism, these superbugs, first of all, won't proliferate because they won't be getting a real good, strong foothold, because you'll be killing them in your own body. Once you get them, and you got a lot of them, and they really take over, then you got a problem. Then you need really strong antibiotics. Yes, I agree with that. I don't disagree with that at all. However, once you take those strong antibiotics, you need to replace all your good probiotics, because you will have killed them just as well. And these, I believe, are the ribosomes, probiotics that you need. And they are just automatically made by living cells. You don't have to do anything. You just live your life and don't disrupt your digestive system by killing the things. And a normal maintenance of the probiotics is that's what I do. It's just what I do. I take them twice a day, and they seem to work. So anyway, we need to do something. What can we do about this global threat to public health? And it's everywhere, and it's mostly because people's digestive systems are not working right. If you're not pooping right, you better get it straightened out. I'm telling you right now, that's the key to everything, is poop. That's the key to everything. And they know this. It's not like it's a hidden mystery. Yale has the thing out that says, eat, poop, and live. All right, this is the most important thing in you, is that you poop right. And Yale University says, eat, poop, and live. Well, why would you eat, poop, and live? And this goes back 8 years ago. This is from 2016. Eat, poop, and live. Why? Why would you eat, poop, and live? Because you're getting that new bacterial stuff in there so you can digest your food. As simple as that. They know about this since ancient times. It's been a common practice. Look at this. This is kind of funny. During a supernova, the star releases very large amounts of energy as well as neutrons, which allows elements heavier than iron, such as uranium and gold, to form. Well, that's what they say. Now, this one says the only way to create substances heavier than iron is by a process called neutron capture, where neutrons penetrate an atomic nucleus. Well, let me show you how it actually does happen, and that's not too far off, but it's done by enzymes. Okay, so this is the atom we're talking about is iron. So how do you get above iron into cobalt, or nickel, or copper, or zinc? How do you get up past that? They say there's no way, except for a super collider or all kinds of things they say. But here's the deal. This is iron, Fe. Now here's the isotopes of iron. They say, again, these are all the protons, 26 all the way down, 26, 26, 26, 26, all the way down. However, these are the neutron values, which I say neutron is nothing more than another figure 2000 particles. It's 1824, I think, but let's just figure 2000. All right, so you got 26 times about 2,000, and you got 19 times about 2,000. You got a bazillion particles here, and they just keep getting more and more particles. So let's say this one, I think I might have shown you this before, but you can't go too wrong here. Now, say this one here is way down here, and we keep adding more and more little particles. It gets getting bigger and bigger and bigger, as it does going down. So in the beginning, if we decay this, we got manganese and chromium and so forth. And then down here, after it reaches stability, we give off cobalt. Well, that's bumping it up. This is a bump up. This process is called bump up. So this is atomic bump up. That's what I'm going to name it. Atomic bump up from iron to cobalt. It is right there. Well, how does the iron break up to decay and get a daughter isotope of cobalt? It's from the enzymes. The enzymes come in and go brrr, and they take a couple off of here, and then they take another one over there and they throw it in here and a couple over there and some of this and some of that, and instantaneously here it is. It's cobalt. It's a bigger product. All right, and that's what they are. And these are the half-lives. And again, to these enzymes, they work like at the speed, literally at the speed of magnetism, which is just instantaneous. Because all they have is magnetism to them, and they're making cobalt. Well, we just saw that cobalt can bump up and go to nickel. Nickel can go to copper. Copper can go to zinc. Simple as that. That's the bump up. And you can go all the way up to uranium, go as far up as you want. There's going to be a bump up. All right, this is important to understand. Transition metals carry ligands with them. Ligands can be nucleophiles, which basically they are all in the bloodstream. It depends on the ligand and the metal involved. They're talking about all kinds of different complex things. I'm talking about blood metals carrying ligands. All right, this is where the enzymes work. Catalysts by transition metals, that's the enzymes, provided an attractive solution to a variety of important problems in organic synthesis, which means how you build molecules, including nucleophilic substitution. This is what I talk about all the time. It's a generalized pathway for palladium catalyzed cross-coupling of an electrophile is outlined in figure 6. Well, they're just showing something. But I can tell you how it does happen. They just bump up. They bump right up. All right, this is important to understand. We have enzymes and then we have coenzymes. They work together. And enzymes are very fast. Some enzymes, like carbonic anhydrase, can catalyze over 1 million reactions in a second, and by doing so they can speed up the reaction rate billions of times faster than at normal rate. Enzymes are generally much better catalysts than inorganic substances. Well, enzymes are created by bacteria, so they are organic in nature. They are in nature. Okay, they understand that enzymes make everything function. They just have no idea where they come from. So how does your body speed up these important reactions? Answer is enzymes, yes. Now how does your body create the enzyme? That's the key. They think it's just your organs, your pancreas and all these other ones create certain enzymes. They just create them automatically. No. They are the mechanism that houses the bacteria that creates the enzyme. Bacteria create the enzyme. That's it. That's the end of the story. Every one of them has its own different type of a house. In your kidneys, it's going to be different than in your lungs. The bacteria is going to be different because you have the different protection needs. It's going to be different in your large intestine than your small intestine, in your bowel, in your anus. All throughout your body, you're going to have different chemistry. That's why there's so many different types of cancer. Every one of them is an invasion of a specific type of body membrane. This is not hard to understand. Once you understand, it's easy to understand. Okay, I don't think it's in dispute anymore that bacteria create the enzymes. Well, when you ask AI here, what creates enzymes? Do organs create enzymes? It's yes. Several organs in the body produce enzymes. They don't produce them. The bacteria produces them, including the pancreas, stomach, small intestine, saliva, gallbladder, and liver. Now listen to this carefully. The pancreas is considered the powerhouse of digestion, producing digestive enzymes that break down fats, proteins, and carbohydrates. Producing, they don't produce them. They are the factory that allows the production to be done by the bacteria that live in that factory. And I'll show you, that's pretty well understood now. So don't forget, they're saying the pancreas is considered a powerhouse of digestion, producing digestive enzymes that break down fats, proteins, carbohydrates. These enzymes travel through ducts in the pancreas and empty into duodenum. I can go along with that. The upper part of the small intestine. The pancreas also produces about 8 ounces of digestive juice each day. That's a lot of digestion going on, and that's a lot of enzymes, and that means you have to have a lot of bacteria. So the pancreas must be pretty well saturated with bacteria if it is the one that produces these digestive juices. So in the pancreas you got all kinds of enzymes, it looks like to me. So you must have all kinds of bacteria, I would think. I would like to see if they can take a pancreas and do a bacterial scan on it. What kind of bacteria lives in the pancreas? Now, we know they say that these enzymes travel throughout the body, and I believe that's true. Now, the small intestine releases maltase. Anything ASE is an enzyme. It breaks down maltose, which is malt sugar, into glucose, which is simple sugar. I think it's C6H12O6, but I believe, but we'll have to look at that. They all have a chemical signature. And the more complicated they are, the harder they are to digest. So you need this maltase to break down the maltose down into simple sugar. The body uses glucose for energy. Saliva also contains digestive enzymes. As soon as you start eating something, it starts getting broken down instantaneously. So I hope you understand, enzymes speed up chemical reactions. It's just like rocket ship fast. And the coenzymes make them even more efficient. and especially the B12, which is just unbelievably complex. This is the B12. You know, look at that. Look at that chemical. And that's all it is. Everything is gathered around the cobalt core, which means all of that stuff can do whatever it has to do. And you may need this B12 for dozens of different processes linking up through that upscale, because I think this would be the up. This, well, I don't want to make any guesses, but something is a molecule that creates bigger than iron, which is an up kick. All right, so it's going up from iron up to cobalt. Cobalt will go to nickel. They'll all go up like that. But you need that enzyme. You need all these really and it just happens. It's like stepping up stairway. And I'm going to tell you right now, they always used to say that the alchemists could make gold out of mercury, and I think they could, because mercury and gold are right together. See, Hg is mercury, gold is Au. You could bring mercury down to gold. I'm pretty sure. I don't know how to do it, but down here, the mercury, the daughter is platinum. All right. So we got the mercury can drop down to platinum. All right. So from way up here, mercury, it can go all the way past gold down to platinum. So that's down the downward side, platinum, platinum, platinum. Here's where it goes to gold. So the mercury here can end up being gold, 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 gold. All right, gold, 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 gold. There's gold all over the place. Gold, 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 gold. Then you get into the next one, which is thallium, which is up. All right, thallium. So now you have just made mercury go from mercury down to gold, and looks like there was a lot of possibilities to do that with the right enzyme, I imagine. And you could make mercury go up to thallium with another, a nucleosynthesis means the nucleus is being synthesized and broken down and added up and moved over and shifted and crunched and done this and that. And that's what happens to create these different isotopes. So you're coming up the metal scale. So can thallium go up? I'm sure it does. I think thallium, no problem at all. It goes up to lead. Thallium goes up to lead. Lead's going to go up. They're all going to go up. They sit inside of a range of particles, but when that enzymes invade, they just smash everything to bits and reconstruct it into whatever they're designed to do. But you got a ton of lead coming out of thallium, which is up. That's up. Okay, I guess I'm going to leave it at this, but this is just staggeringly complicated. Every single animal cell in your body is like this, with membranes, ribosomes, and all of that stuff, and nucleus. It's just everywhere. Now, I have some ideas about testing for bacterial analysis and so forth, and we've been working on it now, and if anybody has any connections that can help out, we could use some intervention with the mainstream about this. Even though it's off the charts crazy, I know to the average person they get scared, they don't want to be involved in it because it's just too much to believe. But if something's true, it's true. And when I show you this and you say that's not true, I don't have any confidence in your ability to determine truth from non-truth. I showed you this. Nobody embedded these fibers inside this. This is just literally impossible. Nobody pushed this lip up and this nose back. Nobody would do that. I mean, it is spectacularly amazing, the teeth and everything. I understand that. but it is what it is. And this is the brain move forward. It's just, there's nobody that could stand before me and answer my questions as to why this would be like this deep inside the skull. How would somebody do that thousands of years ago? Which is what they claim was done. Oh, somebody just carved that. No, absolutely not. That is what happened during this exceptional preservation, as Yale says right here somewhere. There it is, right there. Exceptional preservation by silica-rich oceans. Silica, what was that was made of? Silica-rich, exceptional preservations. Yes, absolutely, 100% agreed. And that's what that's made of, silica. And so are my hearts. I know I showed you these about 10 times, I'm sure. That's a heart. It's just basically all turned to silica, yes. A little bit of blood left in it, but that has absolutely none. I'm kind of stunned, to be honest with you, that there's nothing of blood or anything left there. Completely changed. Just very, very clear, clean silica. That's kind of amazing, but it is what it is. If you can make that happen, somebody can do this, I'd like to see it done. And why would they do this with the nose and the lip? It just doesn't make any sense. This is the real deal. All right, so don't forget, some of the most vulnerable areas in your body are your nerve fibers, and that's why they were linking Parkinson's with gut bacteria, same with Alzheimer's, same with autism. They all have a link to gut bacteria, and they claim to have some ideas of what to do about it with certain vitamins. Now, I'm not going to make any claims about that. That's not my range, my area to make claims, but I do know this is what nerve fibers are like, and there's a bazillion fibers in here, and this is what they look like up close. And every one of those fibers has a batch of insulation around it. Where is it? You see this? You see the little tube right in the center of it? These are all nerve fibers. That's what it is. And this is basically these are membranes surrounding like a wire in a house. All right. Now this is the membrane. It surrounds the whole bundle so it doesn't get invaded. But these are the membranes that surround the fibers, obviously highly magnified. So that's the nerves. If you damage that nerve or short it out or whatever, you're going to have trouble. But you have to have the right enzymes to create the right chemistry to keep that thing functioning correctly, or it will short out. And I believe that's the case in a lot of these neurodegenerative diseases, and it's basically based around bacteria. And all of the research articles say almost every one of them says they have gastrointestinal. Well, every one. You show me one that doesn't. These are, they say, they're the side effects of the disease. No, the disease is the side effects of having a damaged digestive system. If you're not digesting correctly, you're not going to get the right products to make the right things in your body. You're not going to have the right chemistry to attack the things that are attacking you. This mucus, right now there's a huge thing about mucus and in the digestive system, which is absolutely required, 100%, very, very, very important. And they're finding that the mucus isn't as good in some people as in others, and they point to that, which I point to that as being the leucine-rich proteins in the loose junctions that have to move and do all this and that, which is your lungs and your esophagus and all that stuff. They need to be sealed, and some of them are opening up. Apparently, that's what they're looking at now, because the mucus isn't sufficient to keep them coated. You see this? I've talked to a lot of people about their digestion, and they say that these people are going to have Alzheimer's. Typically begin with constipation and sleep problems up to 20 years before getting dementia and debilitating loss of muscle control. Now, Previous research found people with Parkinson's also experienced changes in their microbiome long before other signs appeared. So now they want to analyze the fecal samples of 94 patients with Parkinson's and 73 relatively healthy controls and compare the results. And they found that while different groups of bacteria were involved, so there's different types of bacteria in different countries. So some countries had more whatever than some other type of bacteria for some reason in that country. So while different groups of bacteria were involved in the different countries examined, they all influenced pathways that synthesized B vitamins in the body. Well, what does synthesize mean? Synthesize means to make them, to create B vitamins. All right, so these are different groups of bacteria. Well, the bacteria is what creates the enzymes. All right, different groups of bacteria were involved, but they all influence pathways that synthesize B vitamins. All right, they didn't influence pathways, they just couldn't make them, or something was wrong with them, so they couldn't make them, or they make too much of them, B vitamins, whatever. The researchers found that changes in gut bacteria communities were associated with a decrease in riboflavin and biotin in people with Parkinson's disease. So what do you have to do? You got to look up riboflavin and biotin to understand what they are. So what if there's a decrease? Is that good or bad? And then they showed the lack of B vitamins was linked to a decrease in molecules that help create healthy mucus. There's the mucus part. In the intestines, you need that mucus to keep you from getting invaded. They suspect a weakened protective layer exposes the intestinal nervous system to more of the toxins. Yes, I agree with that. We now encounter more regularly. We keep getting more and more toxins in our diets and all that stuff. You can't help it. It's just part of the thing. You need your body to be able to respond to these things. All right. So they showed that the lack of B vitamins and linked to a decrease in molecules help create a healthy mucus layer. You need that mucus layer between you and things that want to get past you, in the intestines specifically. They suspect a weakened protective layer exposes the intestinal nervous system to more of the toxins. These include cleaning chemicals, pesticides, and herbicides. The real killer is the pesticides. Because the herbicides, the herbicides are what kills plants. And what you got is plants inside us, is these bacteria, literally little plants. Such toxins lead to increased nervous system inflammation, blah, blah, blah. And it just keeps going on and on and on. But I'm pretty sure if you get your bacteria straightened out in the gut, it will take care of the particles that you eat, and it will create protective enzymes that will combat things that are trying to attack you. But the first most important thing is you got to get your barrier set up, which is your membrane. And again, I showed a membrane over and over again. And here's what happens when your membrane's no good. It starts attacking, and it just keeps spreading out, attacking, and then all of a sudden it finds a really weak spot right through. And this is the layer that it shouldn't come through, and this is where the enzyme should be here to kill that. If you don't have the right bacteria living in here, it can't kill that. It's really no more, it's just as simple as that. It really is. Okay, let's leave it where we started. A simple solution to Parkinson's, and it looks like all kinds of other diseases as well, not necessarily with this particular type of B12. However, it looks pretty promising that it is such a complex molecule that it could have very, very strong influence on a lot of biological processes. All right, because it is a transition metal complex, and that's what moves everything through your body and does its chemistry. And that baby's got some serious amount of chemistry it can do. And I think I showed you how we can bump up through and bump up and make all these metals, make anything you want. And it's all done by the enzymes. They're the molecular bammers. I mean, it's so fast, it's done at the speed of magnetism, which is the speed of light. Okay, this is what they're saying is a Lewy body. And they say the micrograph of brain cells containing a Lewy body, which is an abnormal aggregation of proteins. Well, let's look at this very carefully. Okay, my friends, listen to this carefully. This is the Mayo Clinic, and this is about dementia with Lewy bodies. Now, he's saying that you can have it, well, just listen to what he says.
But it is very different.
It's also unique in its clinical features. There are some features
Hold on one second, I'm going to come back.
Is very different.
All right, I'm going to come right back.
Comment.
This Alzheimer's disease is obviously the most common. This is also Parkinson's, and they get mixed up between the two. So he's going to talk. Here he goes.
Mayo Clinic neurologist Dr. Brad Bouvet says dementia with Lewy bodies is often misdiagnosed as Alzheimer's disease, but it is very different.
It's also unique in its clinical features. There are some features that are suggestive of Alzheimer's disease, others suggestive of Parkinson's disease, but DLB really is complex.
Dr. Bouvet says the right diagnosis is key because medications that don't work well for Alzheimer's do help some people with DLB.
I'd love to see them do that kind of a study on people that are taking probiotics. After a couple of weeks of taking the probiotics, did that brain scan change? You can see they're looking at something here. Now it looks like this is getting normal. This one here is this one, and then the Lewy body one. I don't know what your scan means. In other words, they were treating somebody and saying, here, you got an issue. You should be like this. You're like this. But if you were a Lewy body one, you'd be something out here. And so it looks like it's more moderate severity Lewy body. This guy probably wasn't as bad. Who knows? All right, so some drugs work with some with the other. Now, I say there are different types of bacteria that they're affecting, so here it goes.
And his colleagues are dedicated to finding answers and treatments for this disease to help improve quality of life for patients and their families. For the Mayo Clinic.
Okay, it's a Mayo Clinic. Now, again, I would love to see him do that brain scan on somebody that was into, a progressively taking more and more probiotics, and see if that is somebody that did a fecal matter transplant. Because these are the things that make the enzymes, and those are the things that create the chemistry, as I showed you very, very clearly. So I'm going to leave it at that. Thank you. Have a wonderful day, my good friends. All right, here we are with the Lewy bodies. Now, these are intraneuronal inclusions. They're found in Parkinson's disease patient brains. And they're linking them to familial Parkinson's disease cases. So they're in the same family, they think. Now, I don't necessarily agree with that at all. They may be eating the same food, drinking the same water, doing all the same things in the same environment, yes. And they may also get Parkinson's disease more often than somebody that doesn't have those kind of eating habits. And, you inherit certain traits. Not only just hands and fingers and your looks and all that stuff, you inherit how your family lives their life. And that could affect this. I'm not going to drag you through this. These are the different types of these Lewy bodies. And they're fibers that could just sort of build up inside these nerve cells and the neurons. I'm not exactly sure. I don't want to say that I know a lot about these Lewy bodies. They seem to be some kind of globs of stuff that are growing inside the cells. They're not being cleaned out. That's the way I'm seeing it. And they get worse and worse as you get older and older because you're getting less and less bacteria working with this, possibly. But for some reason, it takes a very long time for that to happen. It just gets worse and worse and worse. Now, is there a way to reverse it and bring it back? I would think there probably would be. But that's something that you'd have to do the research on. And that requires doing, some kind of studies. And that always requires some money and a whole bunch of red tape and bureaucratic nonsense, which I guess is not nonsense. I shouldn't be so that way. But we need to get this done. This is not something that isn't unknown. This was known for years and years and years and years and years. That these probiotics are the key to the enzymes. The enzymes are the probiotics and the bacteria are the factories that make the enzymes. They're the factories. You don't have a factory, you don't get an enzyme. All right, this is the Lewy bodies. This is dementia with Lewy bodies. Now look at this thing carefully. They think that's some kind of extra thing that's in there. I say that's been invaded, and the membrane's been invaded. If you look at this carefully, that was where the membrane was, and somehow that membrane got invaded. Otherwise, it would look like this. The membrane would be intact, the internal membranes would be intact, and everybody be good to go. But this one has been invaded. Now, where the invasion's point is it coming right out of here, cutting in? It might look like it got torn up here somehow. Maybe this thing tore it up. But it looks like this is the tear, and it's missing off of here, which it would be wrapped around here. It almost looks like that to me. So if you took this, and you took that and wrapped it right around, right up to here, it would be surmounted again. I believe these are basically the same as the other things, only they're torn apart. Like that white thing was just torn off of there. See the white thing? Just torn off. That's what I'm seeing right there. So here's another Lewy body. I think that's being invaded here by something. I don't know what. But what's happening is it's getting infested and swelling up. And this is the membrane. They all have a membrane around them. But these are healthy, and they haven't been invaded and all inflamed and swelling up like this one. But the center looks like it's pretty much dead. And now it's becoming apparently some kind of a glob that's just laying around there. I'm not sure of that or not, if these things come and go. But if they don't come and go, you're going to have to get something in there that could dissolve that. What would that be? I don't know. Could you take these out and culture them and take them out and check them in a petri dish as to different chemistry that might dissolve them? Because once they're there, I don't know, could you live and everything's fine once they're there? Or once they're there, they're an issue and you got to get them out. Or you got to stop them before they get there. So those are the issues that we got to deal with, all right? So here it is right here. Bacteria are the factories that produce enzymes. When the right bacteria are present in the right quantities and the right conditions, they produce enzymes. And they're talking about they can make them much more economically than people can manufacture them. Because you want enzymes, but you want the body to make them. You could take enzymes. Enzymes are not alive. You just sort of eat them and they do their job and then they go away. Now, in your body is, when it has these bacteria, they create the enzymes just all day long for free. You don't have to take any supplements, but you need that bacteria living in you. All right, I'm going to leave it at that. All right, this is it's all about the bacteria, the fascia, the membranes, and making sure you have the right bacteria. That's and we should be able to test these people, and that's what it did say in this article, wherever it was. Anyway, I'm just going to leave it at this. But they said they would like to do a test on people that come in with certain diseases and see what bacteria they're missing. Exactly. But you need a database of what is a normal, it's going to fluctuate a little bit, but there's going to be some sort of standards that you can look at and then adjust this person to the standard. That's the way I would proceed with the medical industry. All right, I love you all. Thank you. Goodbye.